Hello everyone, I am Aruna Punna, working as Junior Lecturer in Botany in Government Junior College for Girls, Bongir. In this class, I am going to teach you about the molecular basis of inheritance with the formation of nucleosides and nucleotides in DNA and RNA, names of bonds in a dinucleotides, schematic representation of DNA and RNA, number of base pairs in some living organisms, and the discovery of DNA structure. In this diagram, I explained only about adenine by taking this example, this nitrogen base. So here, in DNA, all nitrogen bases we see one by one. Adenine plus deoxyribose sugar. When adenine nitrogen base combined with deoxyribose sugar converted into deoxyadenosine. This is nucleoside. When this deoxyadenosine combined with phosphate group converted into adenilic acid. Adenilic acid is nucleotide. In the same way, already this explained by taking this diagrammatic presentation. Now the remainder we will see. Guanine, this is nitrogen base. When this guanine combined with deoxyribose sugar, then converted into deoxyguanosine, this is nucleoside. Again, this deoxyguanosine combined with phosphate group and converted into guanilic acid. Guanilic acid is a nucleotide. And thymine. Thymine is nitrogen base. When this thymine combined with deoxyribose sugar converted into deoxythymidine. Deoxythymidine is a nucleoside. Again, this deoxythymidine combined with phosphate group and converted into thymidilic acid. Thymidilic acid is nucleotide. And the last one, cytosine, when it combined with deoxyribose sugar and converted into deoxycytidine. Here, deoxycytidine is a nucleoside. Again, this deoxycytidine combined with phosphate group and converted into cytidilic acid. This is nucleotides. This is about the DNA. In the same way, we take the RNA. In RNA, how nucleosides and nucleotides are formed. In DNA, the same thing. In DNA, we took deoxyribose sugar everywhere. But in RNA, just we have to take ribose sugar only. So, adenine nitrogen base, when this nitrogen base combined with ribose sugar converted into adenosine, this is nucleoside. Again, this adenosine combined with the phosphate group converted into adenilic acid. Here, this is directly adenosine. But in this deoxyadenosine, because here the sugar is deoxyribose. That is why always deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine like this in the case of DNA. But here the sugar is ribose sugar. That is why no need to use deoxy, directly adenosine, guanosine, uridine and cytidine like this. So when this adenosine combined with the phosphate group converted into adenilic acid, in the same way guanine, this nitrogen base combined with ribose sugar converted into guanosine nucleoside, and this guanosine combined with the phosphate group converted into guanilic acid. This is nucleotide. In the same way, uracil. In RNA, uracil is present. In DNA, thymine is present. So instead of thymine, in RNA, uracil is present. So here, uracil, when this nitrogen base combined with ribose sugar converted into uridine, this is nucleoside. When this uridine combined with the phosphate group converted into uridylic acid, this is nucleotide. In the same way, cytosine, this is nitrogen base. When this cytosine combined with ribose sugar converted into cytidine, this is cytidine is a nucleoside. When this cytidine combined with the phosphate group converted into cytidilic acid, uridylic acid, guanilic acid and adenilic acid, these are all nucleotides. How a nucleotide formation takes place and dinucleotide formation takes place, I will explain by using this diagram. This is nitrogen base, thymine. This is pentose sugar. This is phosphate group. By the formation of N-glycosidic bond, this nitrogen base thymine bind with pentose sugar. And by the formation of phosphoester linkage or bond, the phosphate group bind with pentose sugar. In this way, by the formation of two bonds, one is N-glycosidic linkage and second one phosphoester linkage. By these two bonds, complete nucleotide formation takes place. In the same way, here also adenine, nitrogen base bind with pentose sugar by the formation of N-glycosidic linkage here also, and in the same way this phosphate group bind with the pentose sugar with the help of this phosphoester linkage. In this way, two nucleotides completely formed. Again, these two nucleotides bind to each other by the formation of this bond, that is phosphodiester linkage. What is the difference between phosphoester linkage and phosphodiester linkage? Here, this phosphate group is binding with pentose sugar, only one pentose sugar. That is is why here just the bond name is phosphoester linkage. But here this phosphate group at a time it is binding with two pentose sugars. That is why the bond name is phosphodiester linkage. In this way, by the formation of this phosphodiester linkage, the two nucleotides link together. The schematic representation of DNA. This is general DNA view. This is clear, complete molecular representation of DNA, phosphate, sugar and nitrogen base. Up to here, this is one polynucleotide strand and sugar, phosphate and nitrogen base. Up to here, this is one polynucleotide strand. Between two polynucleotide strands, hydrogen bonds are present. So here, when you observe this diagram, the black colored ones two are present. These black colored ones are strands which are made up of sugar phosphate. That is why this is called sugar phosphate backbone. Here two backbones are present, sugar phosphate backbones. In this only sugar molecule and the phosphate is present. Towards inside of this backbone, nitrogen bases are fixed. Always nitrogen bases projected inside of the backbone. Here also nitrogen bases projected inside of the backbone, not outside, inside only. And here what is each strand has polarity, 5' end and 3' end. What about this? And one more thing, the two strands always run anti-parallel to each other. For example, if one strand polarity is 5' end to 3' end, exactly opposite polynucleotide strand has opposite. Here if it is 5' end means here towards this 3' end. Here 3' end means towards this side 5' end. Then only proper hydrogen bonds formation takes place between these two polynucleotide strands. What about this 5' end? Here you can see this sugar molecule, pentose sugar, it is having 5 carbons. This is first carbon, second carbon, third carbon. Third carbon here number also has been given. This is fourth, this is fifth carbon. To this fifth carbon, phosphate group is attached. After that, nothing is attached to this. It is not continuing with that. So that is why this is completely lost. That is why this fifth carbon, due to presence of the last combined with phosphate, on the name of this fifth carbon, the end is 5' end. And at the same time, in the same way, this is one nucleotide, sugar molecule, phosphate group and nitrogen base. And this nucleotide bind with another nucleotide. Just now I explained about the phosphodiester bond. In the same way, so many nucleotides bind to each other and form a polynucleotide chain. And this is the last, for example, just imagine this is only last sugar because here the third carbon is not continuing with any other phosphate. That is why here third carbon is last one. That is why this end we can call it as on the name of the third carbon 3' end. In the same way here also the fifth carbon is attached with phosphate group. This is only last. After that nothing is attached to this. So based on the name of the fifth carbon we can call it as 5' end. And in the same way here also third carbon is completely end. After that nothing is attached to this. So based on the third carbon, the end name is third' end. So in this way, what I said, nitrogen bases always project inside of the sugar phosphate backbone. Here this is sugar molecule, this is first carbon. Always the nitrogen base attached to the first carbon of sugar molecule. This is first carbon, this is first carbon. In this way it continues. And here also this is first carbon, to this first carbon nitrogen base is attached. In DNA, how many nitrogen bases are present? 4 nitrogen bases are present. Adenine, guanine. These two are purines. And thymine, cytosine. These two are pyrimidines. Always purines attached with pyrimidines. And one more, always adenine attached with thymine. Guanine attached with cytosine. If here towards this side thymine is present means exactly compulsory towards this side adenine must be present. And here cytosine is present means towards this side guanine is present. Here guanine is present means here cytosine is present. For example, here adenine is present means here where thymine is present. Here, why I represented the blocks some are smaller and some are larger means because adenine and guanine purines are larger in size by having double ring molecular structure. But in pyrimidines, thymine and cytosine, they have single ring molecular structure. That is why thymine and cytosine I represent small blocks, and adenine and guanine I represent with large blocks. Always between adenine and thymine, 2 hydrogen bonds are present. Between guanine and cytosine, 3 hydrogen bonds are present. In this way, by showing antipolarity and by forming the proper hydrogen bonds, by showing 3' end and 5' end and sugar phosphate backbone, so this DNA is always very stable. What about RNA? This is RNA schematic representation. In RNA, only one single strand is present. So this is ash color sugar phosphate backbone. So here, to give clarification, I circled each nucleotide with blue color. So sugar molecule, phosphate group, and one nitrogen base. Nitrogen bases are 4: adenine, uracil, guanine, and cytosine. So here, due to presence of just only one single strand, only directly to this sugar molecule, adenine, sometimes adenine may be present, sometimes uracil may be present, guanine, sometimes again cytosine, again guanine, again uracil. In this way, to this sugar molecule. So here sugar molecule is enlarged. This is first carbon. So always to the first carbon, nitrogen base is fixed. This is second carbon. This is third carbon, fourth carbon, and fifth carbon. Here also you can see the 5' end and 3' end. Here the fifth carbon is attached with phosphate group. This is completely lost. Nothing is attached to after that. And here also this is a third carbon. This is completely lost. After that nothing is attached to the continuation of this polynucleotide strand. In this way, due to joining of many nucleotides, polynucleotide chain formation takes place. The number of base pairs in bacteriophage, pi into 174. This is the name of the bacteriophage. In this, the number of base pairs, short form BP, 5386. This is the diagram of bacteriophage, pi into 174. Already I taught you the number of base pairs is depend on the size of the living organism and their number of characters. What is base pair? Here in DNA, between adenine and thymine, 2 hydrogen bonds. Between guanine and cytosine, 3 hydrogen bonds are formed. Each set we can call it as nitrogen base pair. So here TA is one base pair, CG one base pair, GC one base pair, TA and one base pair. In this 4 base pairs are present. Each base pair is equal to 2 nucleotides. One nucleotide from one strand and another nucleotide from another strand. So here in this way, see here in bacteriophage lambda, this is another type of virus. Here this bacteriophage is also one kind of virus. Bacteriophage means the viruses which attack the bacteria. Different names they have. This is another bacteriophage lambda. In this the number of base pairs is 48,502 because here the base pair number increased. So comparatively this virus is somewhat larger than this. So if the living organism size increases, automatically the number of base pairs increases. And along with not only size, if the living organism have more characters, automatically the number of base pairs is also more. And Escherichia coli, this is one kind of bacteria. It has 4.6 into 10 to the power of 6 base pairs. So this is Escherichia coli bacteria. This is somewhat larger than the viruses. And in human being, the diploid content of human DNA, that is 46 chromosomes. In this, the nitrogen base pairs are 6.6 into 10 to the power of 9. What is diploid content? In each and every human cell, 46 chromosomes are present. 23 chromosomes come from mother and the remaining 23 chromosomes come from father. So each 23 set we can call it as haploid content. So here 46 means diploid content. So in each and every cell, the total number of base pair is 6.6 into 10 to the power of 9. How the discovery of DNA structure? DNA is an acidic substance which is present in nucleus. This is first time identified by Frederick measured in 1869. And this acidic substance named it as nuclein. At the time, that acidic substance not called as DNA, just named it as nuclein. On those days, due to lack of more technical knowledge, scientists are unable to separate DNA and unable to study on molecular structure of DNA for a long time. After some period, Maris Wilkins and Rosalind Franklin, both of they produced X-ray diffraction data. With the help of this one, we can identify the molecular structure. And another observation of Erwin Chargaff double-stranded DNA. What he observed means in DNA, the ratio between adenine and thymine and between guanine and cytosine are constant and equal to each other. How? So here you can see this is DNA. What I said, always adenine bind with thymine, guanine bind with cytosine. For example, this strand is having two thymines are present. Automatically, thymine always bind with adenine. If the number of thymine is 2, so automatically the number of adenine is also 2. Equal. Adenine 2, thymine 2. And at the same time, cytosine is only 1. So automatically cytosine always bind with guanine. If the number of cytosine is 1, automatically the number of guanine is also 1. Here, in this strand, guanine is present. So guanine bind with cytosine. If guanine number is 1 means cytosine number is also 1. In this way, for example, in DNA, two strands are present. In one strand, the number of adenine is 100 means automatically in this strand, the number of thymine is 100. For example, in this strand, the number of guanine is 75 means automatically the number of cytosine is also 75. Like this, between A and T, adenine and thymine and guanine and cytosine, the ratio is exactly equal. So that knowledge has been given by Erwin Chargaff. Based on these researches, X-ray diffraction data and Erwin Chargaff's double-stranded DNA analysis, the ratio between adenine and thymine and between guanine and cytosine equal. Based on these researches, in 1953, James Watson and Francis Crick proposed a very simple and popular famous double helix model for the structure of DNA. In the next class, I will continue this lesson, molecular basis of inheritance, with the salient features of DNA double helix structure, central dogma in molecular biology, how to calculate DNA length and number of base pairs, packaging of DNA in prokaryotes and eukaryotes, nucleosome structure, non-histone chromosomal proteins, and euchromatin and heterochromatin. The 2 marks and 4 marks question and answers of this lesson in English medium are in the description. If you like this video, please share and subscribe to my channel. Thank you.